Good morning everyone, and welcome to today's worked exam answer video. Today I'm having a look at a biological chemistry question which is focused around amino acids, and this is a really representative question of the different skills that you might be asked to demonstrate in relation to the amino acids part of that biological chemistry topic. So we have initially been given a tripeptide, and we're told that that's made from 3 amino acids: glycine, threonine and lysine. With the exception of the first question, which is focused on the tripeptide as a whole, we're then asked 4 questions about the individual amino acids, and these skills: drawing of a zwitterion, drawing the structure of the species at either a low pH, as here, or a high pH, naming using the IUPAC naming rules one of the amino acids, and then thinking about how an amino acid might react in a way that we've seen elsewhere in the chemistry specification. These are all really common things that you'll be asked to do. So I thought I'd have a look at this today because I know a lot of Year 13 students will have either just covered this or be moving on to this in the near future. And I know that not everyone who studies chemistry does biology, and the biologists tend to find this topic a lot easier because they've met amino acids and protein structures and DNA in biology. So let's dive in and have a look at the question. So as I've already said, we have got 3 amino acids which have been joined together to make a tripeptide. And those 3 amino acids are glycine, which is on the left. We've then got threonine in the middle. Don't know if I'm saying that right, but that's what I'm going with. And then we've got lysine on the end. And when we've joined them together to make a tripeptide, we've made what in biology is named a peptide link. But in chemistry, it's exactly the same as our amide link. So the C double bond O next to the NH, that is the amide link or a peptide link. They're exactly the same, but there are 2 names for the same thing. We tend to call them peptide linkages when we're thinking about joining amino acids together. If we were making a polyamide, we would be probably calling it an amide link. And we can see that that's happened between the glycine and the threonine. And we can see that that's also happened between the threonine and the lysine as well. And when we are forming these peptide bonds, we need to be removing atoms and eliminating water. So this is an example of a condensation reaction. So the glycine will lose the OH from the carboxylic acid group. The threonine will lose a H from the amine NH2 group. That will form water, and then we'll get the peptide link formed there. On the other end of the threonine, the C double bond O has lost its OH from that Cu group, so we're left with just the C double bond O. From the lysine's amine group, lost a hydrogen, that gives us the water, and then they come together to give us the peptide link there. Exactly the same as an amide link if you're not a biologist and you've not seen this before. The little dashed lines there, really handily, are showing us kind of where those 3 amino acids have been joined together. And for the questions B, C, D and E, all of them are thinking about the amino acids after the tripeptide has been broken down back into the individual amino acids. So it might be helpful if you're not a biologist, or even if you are, just visually to actually redraw out these structures with the OH going back onto the C double bond O groups and the H is going back onto the NH groups, so that we can see those amino acids in full. Glycine and lysine are on the data sheet, I believe. Threonine isn't. And it just might help you with some of these questions. Not something you have to do though. So thinking about the tripeptide altogether, initially it says draw a separate circle around each of the asymmetric carbon atoms in the tripeptide in the figure. Now there may be more than one asymmetric carbon, and when we say asymmetric carbon, that's another way of saying chiral carbon. So essentially we're looking across the whole of our tripeptide and we are looking for as many chiral carbons as we can find. So a chiral carbon or an asymmetric carbon is one where the carbon is bonded to 4 different atoms or groups of atoms, and anytime we have a carbon that has a double bond to something, that immediately cannot be chiral, because we count the double bond to the same atom as the carbon being bonded to, for example, the oxygen twice, which means that it's no longer bonded to 4 different things because 2 bonds go to the same atom. So that immediately rules out the carbons of the C double bond O's. There's a few others that we can immediately rule out as well, like the middle carbon in the glycine, because that's got 2 hydrogens on it, so that's not chiral. All of these carbons with their 2 H's on them, they can't be chiral either because they've all got 2 H's on them. That carbon up there, 3 hydrogens bonded to it, that can't be chiral either. But there are going to be, thinking 3 at this point, having a quick scan along. So not that one, not that one. This one should be chiral, I would think, because we've got a hydrogen down there, we've got a CHOH and a CH3 up there, and then this way we've got the NHCO, and then because the amino acids are different on either side of the threonine, that is going to be chiral, so we need to put a circle around that. The one above with the CHOH, so the hydrogen coming off one side and the OH coming off another, CH3 above, and then all of this underneath, that carbon will also be chiral. So that's 2. Not that C up there, not the C double bond O there. This is, I think, the 3rd one, because we've got a hydrogen, we've got a carboxylic acid group, we've got the CH2s and the NH2 in the side chain, and then we've got all of this. So that will also be a chiral carbon or an asymmetric carbon. Not the C of the carboxylic acid group because we've got the double bond to the O. Not those carbons because they've got 2 hydrogens on each of them, and then we're done. So it does say around each of the asymmetric carbon atoms. It might have been 1, it might have been more. It's only 1 mark, but that doesn't mean that there was only going to be 1. So make sure you check through the whole structure. Okay, right. Now looking at the individual amino acid questions. So we've got to draw the zwitterion of glycine in part B. So like I said, what it might be helpful to do is to first of all just draw what glycine would look like once this tripeptide has been broken down. So I'm going to answer these questions down the bottom here where I've got a bit more room. So the first thing I'm going to draw is just glycine as an individual amino acid. So nothing changes up until here, where once the tripeptide has been broken down, that's going to then reform into our Q group. So that is glycine, not as a zwitterion, but just separate from that tripeptide. And then what we need to know about zwitterions is that they form when there is an internal acid-base reaction in the amino acid. So the hydrogen of the Q group gets donated, so the carboxylic acid group is acting as an acid, and it donates the hydrogen to the NH2 group on the other side, which is acting as a base. That will then give us our zwitterion. So we will end up with there being 3 hydrogens on the nitrogen, and because the nitrogen is then bonded to the carbon, nitrogen forming 4 bonds, it's using its lone pair in the bond to the hydrogen that it's gained, so we need to give the nitrogen a positive charge. Middle carbon remains unchanged, and then the Q group will now be C double bond O, C single bond O, but that O lost its hydrogen. The hydrogen will leave with no electrons. It's been donated as a proton, a H+, so the oxygen will now be a minus. And that is our zwitterion. So zwitterions are always the H from the Q group moving across to the amine group to give us that NH3+. So that is the answer that they would be looking for. And drawing zwitterions is a really, really, really common thing that they will ask you to do in relation to amino acids. So there we go for that one. As always, any questions, please do let me know and I'm very happy to answer them. Part C, we're sticking with glycine for now, and it says draw the structure of the species formed when glycine reacts with an excess, keyword, of bromomethane. So this is one of those questions where we've looked at reactions of amines with an excess of bromomethane, and because all amino acids have an amine group in them, we might be asked to apply that knowledge to specific amino acids, and the reaction is no different. It's just important that we can recognise that an excess of bromomethane, we're thinking about nucleophilic substitution, and because we have an excess of the halogenoalkane, and that's why that is a key word, an excess of halogenoalkane will give us successive nucleophilic substitution. So we're not gonna just get one reaction of the amine group, we're gonna get it happening again and again and again, because the nitrogen will keep its lone pair. So once it becomes a secondary amine, it can react again to form a tertiary amine, and then again to form a quaternary ammonium salt. So the excess part is key here. So we're going to get successive nucleophilic substitution using the amine group of the amino acid, and that is going to become a quaternary ammonium salt. So what's that going to look like? The rest of the amino acid will stay unchanged. So this was my original glycine structure. So the nitrogen, which we will come back to and tweak in a second, does still need to be bonded to the middle carbon with the two hydrogens and the Q group, and that is not changing. None of that is reacting with the excess of bromomethane. The lone pair, as I said, on the amine group is then what will attack the halogenoalkane. So the nitrogen will attack the delta positive carbon, and then the bond between the carbon and the bromine will break, and that means that the methane group, which will obviously be a methyl, CH3Br being the formula of this. If the nitrogen is attacking that carbon, that means that the nitrogen will then be bonded to the CH3, bond between the carbon and the bromine breaks, bromine leaves as a bromide ion. And that process gets repeated over and over again. So initially we'll have one CH3 group on the nitrogen and one hydrogen, but then the nitrogen still has a lone pair, so then it will attack another delta positive carbon, which will give us another CH3 on here, but in this version the nitrogen will still have a lone pair, so it's going to attack a third molecule of bromomethane, and then we'll get another CH3 on there like that. The nitrogen, again, it's forming 4 bonds, so it will need a positive charge. Now, nitrogen's not a massive fan of being like this. If it is going to be like this, there needs to be some benefit to it having 4 bonds, and just like in the case with the zwitterion, if we have amino acids in the form of zwitterions, we can have ionic based interactions between the positive charge on one zwitterion and then the negative charge on another zwitterion. So if we had another one of these next door here, this O- would attract the N+, and that increases the stability of the kind of overall molecule. Same thing is true with our quaternary ammonium salts. We've got this nitrogen which is positively charged, but the bromide ions will be Br-, and again there'll be that interaction between positive nitrogen and the negative Br, which we wouldn't need to draw because we're looking at the species formed from the glycine reacting in that way. Mark schemes will probably put it in brackets, as in it may be there but it might not be, and it's not really necessary. I'm going to put it there so that you know that that is kind of a part of the quaternary ammonium salt, and that's what enables it to exist in that form. Okay, that is really commonly asked because every single amino acid has an NH2 group. Likewise, every single amino acid has a carboxylic acid group, a COOH, so every single one would be able to react with an alcohol and become an ester. So that's another fairly common question. Other reactions might link into, the functional groups found in the side chain. So if we've got an OH in the side chain, technically that could be oxidized. If it's a secondary alcohol like it is here, that would then give us a ketone. If it was a primary alcohol, then it might give us an aldehyde or carboxylic acid. So these are all reactions we've met elsewhere, but we're applying them in a slightly unusual way to amino acids, but the reactions are always the same. Okay. Next up, we've got to deduce the IUPAC name of threonine. So obviously threonine is the biological name for this particular amino acid. We want to name it based on its chemistry. So again, the first thing that I'm going to do is draw that out. Not in the tripeptide, but just draw out the amino acid of threonine. So we would have H2N, because the hydrogen will go back onto the NH to give us back our amine group. I'm going to flip and put the hydrogen up there because I don't have as much space as I maybe should have left myself. Then we've got the CH and the OH, and I'm purposely drawing this out to try and help me with the naming process in a second. And then we've got the carboxylic acid group there, which again, it's C double bond O. When we hydrolyze the tripeptide, the OH will go back onto that carbon to give us that COO group. That is what we now need to name. And we've got 3 different functional groups in this particular amino acid, which is why they've asked you to name this particular one. It would have been way easier to name glycine because we don't have a long side chain. But it's not impossible to name this. There's a couple of things that we need to, I guess, know to be able to name it correctly. One is that the carboxylic acid group will always be a part of the longest chain, and it will always take priority when it comes to naming and appearing as the suffix of our name. So this will end with oic acid. We can't prefix the carboxylic acid and put it at the start of the name. So we always have to find our longest chain that contains our COO group. So we're going to be going from there, and then because we've got carbons in the side chain, we're going to be going up here like this to find our longest chain. So that's one of the things that we need to know. We will then be prefixing the amine and the OH. They can both become prefixes, amino, hydroxy, but we can't prefix the COOH, so that needs to go at the end. The other thing we need to remember to get this right is that because the carboxylic acid group has to always be on the end of our chain, because you can't have a carbon that's forming 2 bonds to an oxygen and a 3rd bond to an OH being anywhere other than the end of the chain, because it's already using 3 bonds. It couldn't be in the middle of a chain, because then overall you'd have 5. So that means that we never put a number to show that the carboxylic acid group is on the end, but that therefore means that we need to say where all the other functional groups are in relation to the carboxylic acid. So the carboxylic acid carbon will always be carbon 1. And that's the same for aldehydes, which are always on the end of the chain. Acyl chlorides, also always on the end of the chain. Amides as well. But in particular, for this amino acid kind of topic, yes, carbon 1 in your carboxylic acid, carbon 2, carbon 3, and carbon 4. And had we have numbered it the other way, we would get the numbers in our name wrong. And that's why it's important that we know that. Now we can put the whole name together. So I personally would always then name the longest chain bit first at the end, and then do the prefixing before that. So we've got 1, 2, 3, 4 carbons as a carboxylic acid. So the ending of our name is going to be butanoic acid. Then we can think about the prefixes, and you don't have to do the ending first, but I just think it makes sense to, because then you're not trying to think of the whole name in your head. You've got that bit written down. Now we can focus on the substituent groups. So we've got the amine group on carbon 2 and the alcohol group on carbon 3. This will become an amino as a prefix. This becomes a hydroxy as a prefix. And whenever we've got prefixes, it's not that we do it in number order, it's that we do it in it's not that we do it in numerical order, we do it in alphabetical order. So amino comes before hydroxy, because A before H in the alphabet. So this is going to also be in numerical order, but if you had your amino group on a higher number, the amino would still come first in this scenario. So we're going to need 2-amino, 3-hydroxy butanoic acid, and that would be the full name of threonine. Hopefully that makes sense, but as I've already said, let me know if you've got any questions. I'm very happy to answer them. Okay, and then finally, this is another really common question. We're drawing the structure of the species formed by lysine at a low pH. The alternative would be to draw something at a high pH. When we're thinking about low or high pH, we're thinking about acidic or alkaline solutions respectively. So low pHs, pHs 0, 1, 2, possibly 3. We're thinking about placing our amino acid into an acidic solution. An acidic solution will provide H+ ions because acids are proton donors. So what we need to do is protonate the relevant functional groups with some H+. So again, first thing I'm going to do for part E is just draw out the structure of lysine independently of it being a part of the tripeptide. So it would become H2N, CH, and then the COOH, and then nice long side chain for this one. We've got the 4 CH2s, and then we've got an NH2 up there. So when we think about protonating, my head says protonate everything, and that's because I know what I'm referring to when I say protonate everything. When we're protonating amino acids at low pH, what we want to be doing is turning any NH2s into NH3+, because the NH2s are bases, weak bases, but good weak bases, so they will be able to accept protons. So all your NH2 groups will be protonated to NH3, and all of your Cu groups should be Cu groups with hydrogens on, because again, if we're thinking about zwitterion formations, the H will get put back onto the COO-. So if you are going from like a zwitterion to this structure of lysine at low pH, we will want to be putting the hydrogen back on there as well. Now, the reason why they've done this with lysine is because lysine has another NH2 group in its side chain, and we have to protonate both. So if you were to just protonate the main amine group, you would get this question wrong, because it's going to protonate this one as well, because all NH2s are good bases and can accept protons. Likewise, if we were doing it at an alkaline pH or a high pH, we would need to deprotonate every carboxylic acid group, including any that appear on the side chain, and aspartic acid is the most common one they'll ask that about, because of the fact it's got an extra Cu group in the side chain. So the structure of the species formed by lysine at low pH would need to look as follows. And again, based on where I've drawn that, I am going to have to now put the H up there and, really sorry, kind of flip it so that the side chain comes down, because I drew it in a bad place. And then this NH2 down here will also be protonated and give us NH3+, like that. All of those really common skills, all of those things that I would highly recommend that you feel comfortable being able to do. Other questions I might ask: different amino acids at high pHs, naming other amino acids. So I've seen them ask you to name lysine before using the IUPAC rules. Other reactions like ester formation from the carboxylic acid group in any amino acid, or reactions of components of the side chain. or it might get you to, from the amino acids, think about drawing dipeptides or tripeptides like we've got here. So those are other skills that you might want to also have a look at. Hopefully this video has been nice and helpful for you guys, and thank you so much for watching.